Everybody, thanks for coming. So Leanne's here to share with us about viruses and frozen berries and what's going on in the current status of the industry and challenges with FDA. So,
cool. Hi. It's nice to see some students here too. That's really great. For those of you who don't know me, my name is Leanne Jaykus. I'm a faculty member at North Carolina State University in Raleigh. I've been in food microbiology for many decades. I'm probably best known as a food virologist. I was brought into this issue mostly through the American Frozen Foods Institute when this berry situation began to manifest. But I have many, many years of working with viruses in lots and lots and lots of foods, and I have a very soft place in my heart for the berry industry for a number of reasons. So what I'd like to talk about today is kind of a comprehensive look about what's going on. I'm going to give you some history and just put everything into context so that at least maybe there's a little bit more clarity relative to the situation. So by way of outline, I'm going to give you some background information about viruses for those of you who are not terribly familiar with viruses. I'm going to talk about the sampling plan, some of the testing methods, and then I'm going to talk about some various issues related to this that are pertinent to public health, food microbiology, economics, and consumers. What are some of the impacts with conclusions, and then I hope to have about 15 minutes left for questions. So for those of you who are bacteria people, which probably is everybody in this room, we're talking about a completely different beast here when we talk about viruses, okay? So viruses, or enteric viruses, and they're called enteric because they enter through the gastrointestinal tract, and in some cases they cause GI disease, in some cases they cause hepatitis, but the entryway and the infection route is through the GI tract. They're a functional, not a taxonomic group, so there's many different types of viruses that cause enteric disease. A couple of major things to keep in mind is that unlike many bacterial pathogens that have either animal hosts, think salmonella, okay, or are associated with environmental contamination, so think listeria. Viruses are transmitted, they're very species specific, so these viruses are only transmitted by people, which means they're only associated with a human route of contamination. And that route of contamination is 99 times out of 100 is human fecal material from people who have been infected or from raw sewage that has not been treated, okay? The exception to that is that when people have norovirus illness, they do experience projectile vomiting. And we now know that norovirus is shed in vomitus. And actually, in a vomiting event, it can aerosolize. And when it aerosolizes, it can be, I don't want to say breathed in, but it can be enter the mucous membranes and go into the GI tract and cause illness. But it can also travel distances and settle. So we do have to be cognizant of the fact that vomiting episodes are important sources of norovirus contamination. These guys are obligate intracellular parasites. That's a very fancy way of saying they are not alive, but they require a live host cell in order to replicate. And that live host cell is cells in your gastrointestinal tract. What that means is that they don't divide or multiply in foods or the environment. So what gets in the environment or what gets in a food stays in the food. So that's the good news, actually. The bad news is that they stick around for really long periods of time. And when I say long time, I mean like weeks. So if I were to put some norovirus on this countertop here, okay, I could probably still detect it and it still would be infectious maybe 6 weeks from now. So they stick around for long periods of time. Very simple structure. They have a nucleic acid genome, which consists of a single strand of RNA and a protein coat. And the protein code is really what gives these viruses their resilience or their resistance. Just to keep in mind how virus shedding can be risky, or to the degree to which it's risky. A person who's infected with human norovirus is shedding, certainly in their symptomatic phase, they're shedding somewhere around 1 million to 1 billion virus particles per gram of feces. Okay? It only takes 10 to 100 to make somebody sick. So you can imagine 1 gram of feces contains literally thousands of infectious doses of human norovirus. And 1 gram of feces is not a very large amount. So which viruses are of epidemiological significance? Well, there's actually 4 major enteric viruses that are. Rota and Hep E. Rota is the leading cause of viral gastroenteritis in young children. We have a nice vaccine for it. It's more of an issue in the developing world than it is here in the US, and usually associated with water. Hep E is actually a great concern in the UK, or excuse me, in Europe right now, but we in the US have not gotten on the Hep B bandwagon yet, and I don't think we will for a while. So I'd really like to focus on Hep A and noroviruses. So hepatitis A causes the most severe of human enteric virus infections that are associated with foods. It does cause a self-limiting hepatitis, so it's not like Hep B or Hep C where you get chronic hepatitis. Okay, once you get the hepatitis, you recover and you should be okay. The bad news is that the severity of that disease increases pretty dramatically as we age. So people who get this illness when they're in their 50s, 60s, 70s, and 80s can get really sick. And when we see deaths, those are usually the age groups that we see deaths in. And we do see deaths with hepatitis A. Hep A is a surveillance illness. It's a reportable disease in the United States. So the CDC keeps really great records of Hep A cases in this country. And so it's relatively easy to track down outbreaks. And there is a vaccine available. It's part of the vaccination strategy for young children. So young children in this country now are getting vaccinated for Hep A, and it gives lifelong immunity. So we'll juxtapose that to noroviruses, which cause a very different disease. It's a self-limiting vomiting and diarrhea. Self-limiting means it doesn't last very long. I'm curious, the people in the room, how many of you think you've had a norovirus infection? Yeah, it should be almost everybody, okay? Vomiting and diarrhea, it's kind of like it lasts for 24 to 48 hours. The hallmark symptom is this projectile vomiting where you like vomit across the room. And then you recover. It's what my mom used to call the 24-hour flu, what we fondly called the cruise ship virus. Okay, that's what norovirus is. It actually is the leading cause of viral gastroenteritis and foodborne disease. Just to put it in context, in the US, we have somewhere around 20 to 25 million cases of norovirus every year. That's symptomatic norovirus. There's some evidence that came out about a week ago from CDC saying that there's another, double that, there's another 20 to 25 million cases of asymptomatic illness where people are shedding virus but show no symptoms at all, which we don't know a lot about, but I send that out there to make everybody scared. The proportion of most noro is transmitted person to person, but about 25% of noro cases are actually transmitted by the foodborne route. Unfortunately, and part of this is because we don't have good diagnosis, and part of it is because the disease is so common. This is not a disease that's reportable. It's usually, unless it's a very large outbreak, it's usually not investigated. And there is no vaccine. There's vaccination trials going on, but there's currently no vaccine available. So why are these viruses unique in the world of food microbiology? Well, one thing is that, remember, they're human host only, and we don't have a cell culture model, and we don't have an animal model to propagate them in. And so consequently, we can't grow them and get a culture in the same way that we can with bacteria. Because of that, they're pretty hard to detect. And the strategy that we use, because we can't do cultural enrichment, right? Okay, so the strategy we use is to concentrate and purify the virus out of the food or environmental sample matrix, and then do a nucleic acid extraction, and then go on to a molecular amplification method, and we use reverse transcription, quantitative PCR for that, and then to confirm that the identity of that amplicon using DNA sequencing. Consequently, you're not actually detecting the live organism, right? What you're detecting is this little piece of DNA from that organism. And so there is this current debate, it has been going on forever, about what does detection of the nucleic acid of the RNA, how does that relate to the presence of infectious virus? And we're going to talk about that in a little bit greater detail in a few minutes. I said before, they can't grow in foods. They're extremely environmentally stable. We freeze at -80 to preserve these viruses, and we'll preserve them indefinitely at -80 with no loss in infectivity. Even in the refrigerator, they'll stick around for months and months and months. Low infectious dose, we talked about that. And important is the fact that they are very, very resistant to most food processes, preservation, sanitizers, and disinfectants. CDC guidelines for inactivation of human norovirus on surfaces is 1,000 parts per million chlorine if you have a clean surface, 5,000 parts per million if you have a dirty surface. And we've actually done studies at 5,000 on dirty surfaces, and that's not even enough. They're completely resistant to quaternary ammonium compounds. They are resistant to most commercial disinfectants that are used in the food or by the food industry or even in hospital settings. Hand sanitizers, there's variable efficacy of hand sanitizers, but the kind of then it's based on the product. The formulation makes a big difference. But I think the kind of take-home message is that you can never rely on hand sanitizers to completely inactivate these viruses. Cooking, if it's pasteurization types of conditions, equivalent to milk pasteurization, should work. So pasteurized products should be relatively safe. But for instance, gamma irradiation doesn't work at all. What else do I need to tell you about food processes? Oh, ultraviolet, interestingly, works pretty well, and it's a great disinfectant for water, and they use it a lot in water treatment in part because it's effective against the viruses. Unfortunately, ultraviolet is not a great food process relative to inactivation of pathogens. So I'm going to remind you about the major contamination routes in fresh produce, and this is a slide that's taken from a colleague of mine at NC State who does a lot of GAPS training. So it's water, worker, wastes, and wildlife. Keep in mind, we don't have to worry about wildlife relative to these viruses. Waste would be generally sewage or raw human waste from maybe a child defecating in a field, and we've actually seen that happen in developing countries. Workers, or of course sewage. Workers, usually that's almost always the hands of infected food handlers or pickers in a field, okay, that don't practice good personal hygiene, okay, so they get fecal material on their hands and then they pass it on to the food that they're coming into contact with. And water can become contaminated when there's any type of sewage breach, untreated sewage breach, whether that's a flooding event, sewage overflow, leaking septic tanks, things like that. Okay, so that's where product, particularly relevant to berries, is becoming, or how it's becoming contaminated. So the frozen berry sampling plan that was put forth by FDA was technically started in November of 2018. It was preceded by a year, maybe a little bit more, where it was announced to industry and other stakeholder groups, and it was published in the Federal Register so that comments could be made. The agency is sampling 2,000 samples. 1,000 of those are imported berries, okay, and 1,000 are domestic. And what's being sampled is frozen berries, not fresh berries. But the imported frozen berries might have been frozen in the US, in an IQF tunnel in the US, but their source would have been international, okay, another country. Sample collection is happening in distribution centers, warehouses, and in facilities. At the current time, they're looking for Hep A and they're looking for norovirus. There are 2 genogroups of norovirus that cause human disease, and because norovirus is a pretty genetically diverse virus, that requires 2 different tests. So it's actually 3 tests that are being done. It's just 2 virus types that are being tested. And they're doing, as I said before, RT-qPCR followed by Sanger sequencing. And then this is a statement about if the FDA finds genetic material, usually they let the distribution centers or the retailers know, who then let the processors know that there's a positive, and there's all kinds of negotiations and additional testing as to what to do relative to product disposition. So this is the most recent, and it's clearly a little bit dated, but the most recent information on the results of the sampling plan. And that's the end of September of 2019, so it's almost 4 months old. So as of that period of time, 25% of the domestic samples had been sampled and 32% of the imported samples had been collected. And a total of 10 positive findings were recorded or reported by FDA. Now, I can tell you that since that reporting time, there's been another 2 or 3 that have been reported that aren't included on this chart. 3 of those were Hep A, 3 of these 10 were Hep A, 7 were Noro. And I noted before that they have to confirm using sequencing, and we will talk a little bit more about that in a second. So 6 out of those 10 have been sequenced positive. If you break it down by the microorganism itself, relative to Hep A, the products that have been positive were strawberry, blackberry, and raspberry. And actually, I forgot to mention this. There's only 3 berry types really that are being included in the sampling plan. So it's frozen strawberries, frozen blackberries, and frozen raspberries, whether they're red raspberries or black raspberries. Blueberries, frozen blueberries are excluded, as are fresh berries. And so that's the Hep A data for Noro. It's been strawberries and raspberries. And note for Hep A that all of the positives have been domestically produced product. None of the imported product has been shown to be positive. And for Noro, at least as of this date, for Noro, it's about 50-50. I want to jump into some public health aspects that should kind of be considered as we begin to look at the data that's coming from the sampling plan results and interpret kind of what's going on. And the first thing I'm going to show you is some information on epidemiological evidence from the past 20 years. What are the leading contributors to viral foodborne disease? And then I want to make a note, and I'm not going to discuss this further, but I do want to make a note that no noro or hepatitis illness cases have been associated with any of this recalled product. Okay, so all of the product that was confirmed was recalled. If it was Hep A positive, it was recalled as class 1. If it was noro positive, it was recalled as a class 2. But none of that product has been associated with any illness. And much of that product had been on the market for quite a while, actually, before the samples were even picked up for testing. So what do the last 20 years tell us about outbreaks? And I was fortunate to know, or to find, this article from Nasheri et al., and it's quite recent, where they actually looked at the worldwide outbreaks of noro and Hep A in frozen berries. So it's very specifically for this product. And if you look at the second column, it'll give the location of the outbreak. Remember, these are worldwide outbreaks. And if you look at the right-hand column, I've circled in blue everywhere where it was an imported product that was associated with that outbreak. And you can see here, on this slide here, we had one multi-state US outbreak, product from Turkey. All the other worldwide outbreaks had product from imported, from other countries, but it was all imported product. And then we had another berry outbreak. This was the smoothie outbreak, where the product came from Egypt. But again, the predominance of product that's coming from outside of the country in which it was, in which the frozen product was produced. Very similar situation for norovirus. I circled the German outbreak only because it's the largest foodborne disease outbreak we've ever had. There were 10,000 cases. It was huge. But again, largely imported product, and very little, only one outbreak associated with product that was manufactured in the US. But again, the source of the raw material was China. So common epidemiological themes are that the vast majority of outbreaks in Europe occur in Europe and not in the US and Canada. And there are probably 5 outbreaks that can definitively be linked to frozen berries in the US over the last 20 years. All of those were associated with imported raw material. And the countries of origin that are kind of at risk seem to be Eastern Europe, specifically Poland and Serbia, the Middle East, which would be Egypt and Turkey, and China. So there is no history of seeing viral contamination in frozen berries that were domestically produced. And that includes the US and Canada. I'm bringing this slide up only as kind of a newsflash. We haven't had a lot of association of outbreaks with fresh product. But this outbreak did occur this past fall, and some of you may have heard about it. And I only bring that up because fresh is not part of the sampling plan, but to show you that there has been a recent outbreak of Hep A associated with a fresh product. The FDA has not yet let us know whether that product was imported or domestically produced. So again, historically, where does contamination occur when you have viral contamination of food? So if you look at the right-hand side of this graph, this is from some data from the CDC that shows that most viral contamination of food occurs in restaurant settings, catering and banquet settings. In fact, the vast majority is well more than 75%. Okay, that implies food handler. Okay, that implies food handler. And CDC is pretty definitive that food handlers are the most common cause of contamination, particularly in restaurant settings. This other slide is really interesting, also produced by CDC. in which, this was Aaron Hall did this work, in which he looked at the top products that were associated with norovirus. And this is only norovirus because this was what they had data for, okay? Norovirus illnesses, and found that fruits and nuts kind of categorically together were the second most kind of risky product category. But what Aaron did in his work is he looked at what we knew about the source of contamination. Where did those fruits and nuts get contaminated? And you can see here the light gray part shows an unknown source, so we don't know where those became contaminated. The medium gray shows service and preparation, again implying food handlers. And only a very small proportion are contaminated at production and processing, and that is this teeny tiny bit here. So there's not a lot of precedent for berry contamination occurring at production and processing, even from the epidemiological literature. Okay, what about testing? Okay, I'm going to talk just a little bit about the testing method that's being used by FDA. And some issues with testing in general. It turns out that there are two different methods of extract. Remember we said when you pull viruses out of a food product, you have to concentrate and purify them from the sample matrix. And we call that virus extraction. So there are two different methods of virus extraction. One of them is the International Standards Organization, or the ISO method. And the second one is the method that FDA is using in this sampling plan, as well as for any regulatory actions that are being taken in response to the sampling plan. And I'm going to talk a little bit in greater detail about those two methods. I'm going to also talk about limitations to detection using PCR, because PCR is not a perfect method, particularly for viruses. The need for robust sequencing for confirmation, and the issue of can PCR product actually be used as an indicator for product adulteration. So you can't tell, or at least I can't tell because this is hidden by the upper graphics, but this is the ISO method, and for the student, and I'm going to just go into really briefly show you. You begin with a 25-gram sample of berries. You go through this is the sample concentration or extraction method, and it's a pretty simple method. You use polyethylene glycol, which is a protein precipitant, and it actually allows those the viruses to sediment as if they were proteins. And then you do a quick centrifugation step and a few cleanup steps. Then you go into RNA extraction, and then you go into RT-qPCR and interpret the results. That's the ISO method. And those of you who are writing, I'm happy to share these slides. This is the FDA method. So you start with a 50-gram sample, so bigger sample, okay, double the size sample. The virus concentration, though, isn't polyethylene glycol, it's an ultracentrifugation method. So you're ultracentrifuging at very high speeds and allowing the viruses to sediment through ultracentrifugation. It has a more complex RNA extraction method, and then you go into RT-qPCR and you interpret the results. So two very, very different methods, particularly when it comes to these two steps in the process. So this is just a summary slide of the major differences. We've really only we've already discussed these. I don't need to belabor it. There's also some minor differences between the methods that I won't talk about in this presentation. But what I really want to highlight is the ISO method is what is being used by commercial labs. So if a processor wants to test their product, they are going to get the ISO method. The other thing is that the ISO method costs about $300 per sample. It's a very complicated method. And that's juxtaposed to salmonella testing, which costs maybe $25 a sample. So much, much more expensive. Okay, so this is going to get a little bit complicated for those of you who are not familiar with PCR. But the PCR detection method, okay, is a nucleic acid amplification method. And the way that works is you're looking for a small piece of the genetic material of the virus. And really, we're only looking for about 1/100th of the length of the virus genome. The PCR works as an exponential amplification. So if you have one copy of that piece of DNA, and you do one cycle of amplification, 1 goes to 2, a second cycle of amplification, 2 goes to 4, a third cycle, 4 goes to 8, 8 goes to 16, so on and so forth, until you have enough that crosses a threshold, okay? And what you actually get out of the PCR machine when you're doing this is what's called a CT value, or a threshold cycle value, and that's the cycle number at which you actually can facilitate detection. So the more initial copies of the virus genome you have, the lower the CT. The less copies, the higher the CT. And that's actually shown in this bottom figure here, where you can see the cycle threshold goes, which is basically in fluorescence, becomes much much higher as the number of template copies is lower. Now, in labs such as mine, and internationally when we do this, we look for a linear range of detection. And that linear range of detection, or I should say a log linear range of detection, the log linear range of detection usually is between a CT value of about 10 and 40, maybe a little lower than 40. Once you get above a CT of 40, PCR becomes kind of wonky. It becomes kind of unreliable. And so you have to be really careful in interpreting results at these very, very high CT values, okay? And that is not only my standard that I use in my research lab, but that's more or less an international understanding for those of us who work with PCR, and it's part of the MICU, or I think they're called the MICI guidelines as well. I want to point out that all of the CT, that FDA does not have a CT cutoff. So any positive CT value is a positive, or a presumptive positive. And that all of the samples that have been, all the product that's been recalled to date had CT values over 40. So 42, 44, there was one that had a CT value of 49. To summarize, high CT values should be considered only presumptively positive, okay, not as confirmed. We're only detecting a tiny piece of the viral genome. We know that nucleic acid, okay, can be stable long after a virus or bacteria has died, okay? So there's not a definitive relationship between the detection of the nucleic acid by PCR and the presence of infectious virus. So the take home is that detection by PCR is not assurance that you have a live or an infectious pathogen there. And there's also this issue with infectious to non-infectious ratio, but we don't have a lot of time to go into that today. Because of that, it is essential to confirm the little fragment of DNA that you have detected by PCR. And that confirmation is done by DNA sequencing, okay? And that's really pretty much the only way you can do that. What DNA sequencing does It allows you to assure that you have a wild type virus present, so you have a circulating virus present in your sample, and that you haven't cross-contaminated your sample with your positive control. That's really the two values of sequencing. And still, though, even with sequencing, it doesn't address the infectivity issue. The considerations for sequencing there are pretty standard, those of you who work in sequencing and bioinformatics, there are pretty decent standards now for sequencing and sequence quality, consistency, alignments, how you do the bioinformatics, how you do the alignments, and how you interpret the results. And those need to be followed and followed strictly in order to have adequate, reliable sequencing results. And without high quality sequencing, then there remains an elevated likelihood that you're going to have false positives. And there's some questions as to the quality of some of the sequencing that has occurred in the sampling plan. Okay, this is my favorite as a food microbiologist. So you guys all probably know what microbiological indicators are. They are used as proxy to monitor conditions in an environment or in a product. The one in food microbiology we probably most often think about is E. coli and coliforms, 2 ones. It's really important to recognize that we're in no way ready to use PCR results or a PCR positive signal as an indicator of human fecal contamination. Okay, we haven't validated that. And the reason it's not yet appropriate to do that is because, number one, we're only amplifying a tiny amount of the virus genome. Number two, we don't know if that piece of DNA was associated with infectious or non-infectious virus. We know very little about the persistence or prevalence of small pieces of DNA or RNA in the environment. In other words, do small pieces of DNA from viruses sit around in water for long periods of time? We're starting to actually get some data that's occurring with antimicrobial resistance genes, that's occurring with some of the bacterial virulence genes, but we don't know if small pieces of DNA from viruses stick around in water, let's say. And so the best that we can currently say is if you find the presence of viral RNA, it may or it may not represent contact with human wastes, which is a pretty wishy-washy statement, okay? Okay, I want to jump to some of the economic issues associated with the recalls. First of all, the cost. I think easily it's $500,000 to $1 million for many of these companies, maybe more than that in some instances, depending on the size of the recall. There are always reputation costs. So there's costs to the retailers who frequently are the ones that are implementing the recall in their facilities if the product is in market or was pulled off retail shelves, which happened at the beginning of the sampling plan. There's certainly reputation costs to the producers and to the processors, but there's also reputation costs to the industry as a whole. How safe are frozen berries? Okay. Is this a product I should be serving my family? There are also lost sales. And anecdotally, I was just up at the Berry Commission meeting in Washington State last week, and a couple of the processors said that anecdotally, what they saw after their recalls that there was about a 10 to 11% sales drop that lasted for maybe 2 to 3 months. Now, is this temporary or sustained? At this point, it was probably just temporary. If it continued over a long period of time, we might see that it's sustained. Consumer confidence issues. This, which you guys here on the West Coast didn't see this like we did in the US. This is a Washington Post article that happened after the first 3 or 4 3 or 4 recalls occurred. So it was seen pretty widely on the East Coast. And I want to make a couple of comments about this relative to consumer confidence. We do need to recognize that berries are ready-to-eat food, but they're also increasingly being used as an ingredient in other ready-to-eat foods like smoothies, okay, which can certainly get contaminated by a food handler too. So the sampling plan as it currently stands is directly implicating frozen product, okay? It's not implicating anything associated with a food handler and food preparation. This can be really confusing to a consumer. You know, where is the contamination coming from? And from a confidence and also a sales standpoint, the fact remains that frozen berries are a very healthy addition to someone's diet. They're healthy food. There's food that we should be eating and we should be promoting the consumption of. Okay, so American Frozen Foods Institute, AFI, went to their constituents and got a feel for what was happening on farm and what was happening in IQF processing relative to safety issues or some of the production inputs. And what they found was that for farm production, you're generally seeing gaps and good farm worker health and hygiene training programs. There are a variety of irrigation types that are being used, but by and large, it's drip irrigation using subsurface or really deep well water. And in many, many instances, frozen berries that are intended for frozen processing are being harvested mechanically. Certainly in the Pacific Northwest, they're mostly being harvested mechanically. So that calls into question is where are your risk factors for human fecal contamination if you're using these kind of production variables, okay? And it's very similar in IQ processing or IQF processing. Good water, good treatment, attention to personal hygiene, hand washing facilities, health and safety plans, and incident awareness. And I can say that I have visited in my visit here, how many? Probably 5 plants. And I saw all of this in all of those plants. And I think 3 out of the 5 that I visited have had incidents with recalls. So I'm gonna wrap it up and give some time for questions. If you are a processor and you get a positive result, what should you do? Because keep in mind, we still have another probably 1,300 samples that are going to be pulled. And of that, about 700 of those are going to be domestic. First of all, realize you're most likely going to hear it from the retailer because the samples are being pulled from the distribution houses. So you're probably not going to initially hear about it from the agency. You should immediately request the RT-qPCR testing data from the relevant Office of Regulatory Affairs FDA lab. And they are releasing those pretty rapidly at this point. You certainly should contact AFI because they will serve as an advocate and they can help you get information that you may not be able to get readily from the agency. You should immediately, whether you have a confirmation or not, because what you first hear is of a presumptive positive, okay? You should still begin your own traceback exercise. You should start that process so that you're prepared. You definitely should expect FDA to show up at your doorstep relatively quickly. And consequently, you should have your food safety and traceability documents at hand. And then you should push for timely sequence confirmation by the agency. The agency is pretty much committed to getting the sequencing results out. I think it's within 10 days, but it's 10 days or 2 weeks. And AFI, again, can help you help advocate to get your results fast. So what are the long-term consequences to processors and producers? Well, assuming that this product is contaminated, and I'm not making that assumption because I think there are some significant testing issues, but assuming that that product is contaminated, it's contaminated at a very low level, and given what we know about viruses, okay, it can only be the ag waters, the wash waters, or the hands of pickers or people in the processing facilities. That's the only place this contaminant can come from. With the exception of vomiting episodes that we talked about, but those are pretty rare, but still need to be part of your passive or harvesting plans. So really prevention of virus contamination must be, of course, a consideration in those plans. Unfortunately, aside from pasteurization, there is no good method yet available to inactivate these viruses in frozen berries. There's a lot of debate about the value of testing product. There are a few retailers who are now requiring testing. I'm not a big advocate for that because I think the test methods are not really good enough to be reliable as a management tool. They might be used as a validation tool, but not as a management tool. And even as a validation tool, they're still not really reliable enough. But some of the processors are being required of the retailers to test for product. But keep in mind that if the product is tested, it's using the ISO method. It's not using the FDA method. And FDA says that they have much more confidence in their method than they do in the ISO method. You might consider vaccination of workers for Hep A. That's a very contentious issue, but some people are looking into that. And I think there's a very real possibility, although there have been no announcements to this fact, that frozen berries may eventually fall into a high-risk food category, which means that how you manage that product into the future is gonna change. So conclusions, frozen berries are considered a ready-to-eat product. Keep in mind that there are times that frozen berries do go into product that as an ingredient in other products that are cooked. Think about pies, think about jams and jellies, things like that. Okay, but they're considered by the agency as a ready-to-eat food. While the risk of domestically produced frozen berries causing an outbreak is quite small, based on past history contamination events, if there were a contamination event, it could cause a significant public health risk. As we said before, we got more sampling on its way over the next year. I tried to cover many of the issues associated with the sampling plan and its interpretation. I could not cover all of them. So suffice it to say that this is a very, very complex issue that has a lot of nuances. And in a way, we really are charting new territory from a food microbiology standpoint, as regulatory actions in this case are being taken on a non-cultivable pathogen, a pathogen we can't culture without a culture in hand, and exclusively using molecular techniques with no supporting epidemiology or no supporting reported illnesses. And with that, these are just some proposed guidelines and research areas that AFI has put together in their conversations with industry, and as they're debating on how to move forward in helping the frozen berry kind of manage this situation and get more science behind what may or may not be occurring. And then information for myself and Dr. Sanjay Gamala at AFI. I will tell you that I did a webinar, I think it was in July, on testing. It was about 45 minutes. It's much more detailed than I could provide here. And I did speak to Sanjay yesterday, and he said if anybody wants access to that webinar, just shoot him an email message, and he'll give you that. And it talks about the pros and cons of testing and what the test can and cannot do. And with that, I will wrap up and take questions.
So,
I'll
I
unmute
have, okay,
You have the questions and I'll unmute this.
yes, great. So thank you, Leanne, this has been great. And I
Thanks.
also wanted to say thank you to everybody that joined us online. So we did have 69 participants
Great.
online as part of the Western Regional Center to broadcasting to the group. So I've received quite a few questions, so I'm not sure how much time we have and in addressing them, but I'll open it first to the room, and if you guys have any questions, we probably have time for one or two before we move on to a lot of them here online.
I just was wondering why they excluded blueberries from the sampling.
You know, that's a great question, and we actually don't know. We really don't know a lot about the evolution of and who initiated it or whatever. And I think part of that has been some changes in leadership in the agency. So I don't know the answer to that.
That was also a question.
Okay.
online, so, okay. I'll move to first question we had. Should a recall be initiated prior to DNA sequencing?
Okay, this is so what is currently being done. Okay, so there's this is a complicated question to answer. Okay, initially a recall was being initiated before sequencing, and AFI did go to the agency and requested that sequencing be done before a recall was initiated. And this got complicated because at that time there was product that was being pulled off the shelf of a retail market, and so it was like you need to get it off the shelf. Now product is being pulled in distribution centers or at the processor, and consequently we are waiting for the confirmation results by sequencing before taking action. Okay, so it's not so much a should as much as it is what has historically happened and what is happening. Okay.
Yeah, thank you. There was another question that said, I think you said that all FDA results have been high CT values over 40, yet I think you said that 2 out of 3 presumptive positives have been confirmed by Sanger sequencing. Does that make sense to you?
So it actually was, and this was looking at the September 2019 data, it was 10 total positives, 6 out of the 10 were confirmed by sequencing. And yes, every one had a high CT value, greater than 40. It doesn't surprise me that you would not have things confirmed out by sequencing at such a high CT value, because our experience has been that when you hit a CT value of 40 in our lab, we almost never can confirm it. Okay, so that's not a surprise to me. In my opinion, and this is my opinion in scrutinizing the sheets from the agency, I think that some of their confirmation positives at high CT values may be explained by circumstances other than the presence of wild type virus. And I think that that's kind of where I'll leave that. If that person would like to contact me for further discussion, I'm happy to talk to them.
Great. Do you have any idea what % of the US population has been vaccinated for Hep A?
Yeah, that's a really good question. I don't. I probably could look it up. I can tell you that I think more or less the belief is that that most of the population over the age of, certainly over the age of 40, is still susceptible.
And can someone who has consumed contaminated berries be treated with vaccine to prevent illness?
Yes, as long as it's 2 weeks after exposure. Okay, but one of the problems with Hep A is that it's a 4-week incubation period. So a lot of times before you ever see the first, especially if it's in a restaurant, before you ever see the first case, it's too late to protect by vaccination. But in instances where there have been prolonged outbreaks identified, what retailers will oftentimes do is offer free vaccination to people who return the recalled product.
Has seasonality / time in the growing season been studied as a link to presumptive / positive test results?
No.
That's an easy, quick one to answer. What is the false / positive ratio for PCR, and how is this accounted for in terms of duplicate testing?
Okay, so that's kind of a vague question. Okay, I can tell you that the way in the FDA sampling plan, they take 3 subsamples of the product, and they do an extraction on each of those 3 subsamples, and then they do 3 PCRs on each of those 3 subsamples. So they're basing their results on 9 separate PCR reactions, okay? FDA's stance is that a single positive, so any of those 9 coming up with a CT value, makes that sample presumptively positive. There's many different ways of dealing with this elevated CT thing. Some people set a cutoff. There's other organizations that, I think some organizations in Europe will say, okay, well, if you have positives at high CT, it needs to be 3 out of 10 or something like that. So I think that's probably the best way to answer that question.
Great. And is there a study citing what pasteurization temperatures kill the norovirus or Hep A?
There really aren't, and we've done some studies in our lab. There's other labs internationally that have done some studies. I just generally use a benchmark of milk pasteurization because I'm a teacher and I teach about milk pasteurization, right? So that's 71.7 for 15 seconds or 62.8 for 30 minutes, or the equivalent, and usually there's a margin of safety there. And I only say that because we haven't really seen any outbreaks associated with pasteurized product. So it's a benchmark, but there's not a ton of data that supports that is an appropriate benchmark, but there's not data to support that it isn't either.
Okay, great. And then the last question was, will this presentation be posted online to review again? So we have recorded and we do have plans to share it with through the Western Regional Center. So
and I appreciate everybody for joining.
And that's 2 p.m. So this is perfect timing. So let's please thank Leanne for a wonderful presentation.